Enzymes. Types, functions, and mode of action. Introduction. Enzymes are biological catalysts that play a crucial role in various biochemical reactions. They speed up chemical processes in living organisms without undergoing any permanent change themselves. These proteins are highly specific and work under optimal conditions of temperature and pH to ensure efficient metabolic processes. In this article, we will discuss the different types of enzymes, their classification, functions, and their mode of action in biological systems. What are enzymes? Enzymes are macromolecular biological catalysts that facilitate and accelerate chemical reactions. They are made up of amino acids and have a unique three-dimensional structure that determines their function. Most enzymes are proteins, although some RNA molecules, known as ribozymes, also exhibit enzymatic activity. Properties of enzymes. 1. Catalytic property. Enzymes significantly increase the rate of chemical reactions. 2. Specificity. Each enzyme acts on a specific substrate. 3. Reusability. Enzymes remain unchanged after the reaction and can be used multiple times. 4. Sensitivity to temperature and pH. Enzymes work best within a specific range of temperature and pH. 5. Regulated activity. Enzymatic activity can be enhanced or inhibited by various factors. Types of enzymes. Enzymes are classified based on the type of reaction they catalyze. The 6 major classes of enzymes according to the Enzyme Commission, EC, classification are: 1. Oxidoreductases. These enzymes catalyze oxidation-reduction reactions, where electrons are transferred from one molecule to another. 1. Example: lactate dehydrogenase, converts lactate to pyruvate. 2. Transferases. Transferases facilitate the transfer of functional groups between molecules. 1. Example: alanine transaminase, transfers amino groups in amino acid metabolism. 3. Hydrolases. Hydrolases catalyze the hydrolysis of chemical bonds, breaking them down with the addition of water. 1. Example: amylase, breaks down starch into maltose. 4. Lyases. Lyases catalyze the breaking of various chemical bonds without hydrolysis or oxidation. 1. Example: aldolase, involved in glycolysis. 5. Isomerases. Isomerases facilitate the rearrangement of atoms within a molecule. 1. Example: phosphoglucomutase, converts glucose 1-phosphate to glucose 6-phosphate. 6. Ligases. Ligases catalyze the joining of two molecules, usually with the expenditure of ATP. 1. Example: DNA ligase, joins DNA fragments during replication. Mode of action of enzymes. Enzymes function by lowering the activation energy required for a reaction to proceed. The process involves several key steps. 1. Substrate binding. The substrate binds to the enzyme's active site, forming an enzyme-substrate complex. 2. Formation of enzyme-substrate complex. The enzyme undergoes a conformational change, facilitating the reaction. 3. Catalysis and product formation. The reaction occurs, converting the substrate into the product. 4. Product release. The enzyme releases the product and returns to its original state. 5. Enzyme recycling. The enzyme is ready to catalyze another reaction. Factors affecting enzyme activity. Several factors influence enzyme activity. 1. Temperature. Enzymes work best at an optimum temperature. 2. pH. Extreme pH levels can denature enzymes. 3. Substrate concentration. Higher substrate concentration increases the reaction rate up to a saturation point. 4. Inhibitors. Substances that decrease enzyme activity, such as competitive and non-competitive inhibitors. Applications of enzymes. Enzymes have wide applications in various fields. 1. Medicine. Used in drug formulation and disease treatment. 2. Food industry. Employed in baking, brewing, and dairy industries. 3. Biotechnology. Used in genetic engineering and biofuel production. Conclusion. Enzymes are essential for life, facilitating biochemical reactions in living organisms. Understanding their types, functions, and mechanisms helps in various scientific and industrial applications. Their specificity, efficiency, and versatility make them indispensable in biological systems and technological advancements. Subscribe.